So whilst the few remaining ones are entering the room, we will indeed start the session, Saving European Species and Biodiversity Together, which is of course linked to EAZA's new vision, Progressive Zoos and Aquariums, Saving Species Together with You. Yesterday, for those of you that were in the opening session, might have heard the opening and welcome address from the European Commissioner for Environment, and I need to double check the name so I say it correctly, Mr. Sinkevicius, and he really showed that the EU is wanting to be a leader globally to help get to a biodiversity framework following the decade of biodiversity that is ambitious and that is really going to lead to transformative change. And as a European, and also hearing what goes on elsewhere in the world, that makes me really pleased to see such ambition and such leadership. And also the recognition that nature has its own intrinsic value as well as an important role for humankind, and that humans are part of the biodiversity. So that was a really pleasing message to hear. There is a European biodiversity strategy that is linked to the Kunming CBD next decade, or post-2020 targets rather. And EAZA has developed a position statement to the EU biodiversity strategy. And two things that we call the EU on is that we are pleased, of course, to see the ambition, but that we also really would like to see that ambition is turned into daily practice. And that, of course, is not only the European Commission, but we also hope that the Parliament and the Member States show an equal level of ambition. And we really are keen to see the role and the full potential of zoos being recognized as a partner in some elements of helping restore and maintain biodiversity. And in that context, both from an EU and a zoo side, it's only it is really relevant to look at the work we are doing for European biodiversity towards saving species in our zoos, working with partners. And to also explore if there is further potential for us in this area. So with that introduction and without further ado, I would like to introduce our first speaker, which is Maurice La Haye from the Zoogdierenvereniging, as well as the Radboud University in the Netherlands, who will be presenting on what is, I think, over a decade of work on one of my favorite species, the common hamster. So Maurice, if you're ready to share your screen, then the floor is yours.
Yeah, perfect, thank you. So, thank you very much, Danny, for the introduction. Colleagues, welcome. Thank you very much for the invitation to give this presentation about the common hamster in the Netherlands. I'll give you an overview of almost 2 decades of hamster research on in the Netherlands. We started in 2002, well, even a few years earlier in 1999, we started with the conservation of the common hamster in the Netherlands. I will give you an overview of the project of the last 20 years. I hope everybody can follow me and can hear me. No problems? Yeah, perfect. Yeah. Common hamster, well, it's a very nice, colorful rodent. It's also my favorite species, so we share some favorites. It's a typical species of the agricultural landscape that is in the Netherlands. It's original steppe species, and it made a successful step to the agriculture landscapes in Western Europe. It's quite a large rodent, a few hundred grams, living and burrowing on the ground, mainly on arable land, and it can be found mainly on less or loamy soils, also on other more soil in the western part of Europe, which I don't know, but the Netherlands it's less. It was placed on the IUCN Red List of critically endangered in last July. It has for years been on the list of the IUCN as least concern because it's a widespread species all over Europe. It has a distribution until the western border of China. But well, now we see globally a large decline of species, and all kind of populations are going extinct at the moment. I'll show you a map of Western Europe. The part that is Netherlands, and it's only living in a small part of the south of Limburg, but that has a large distribution in the whole of Europe. The pink area is the former area where it used to live, and this is a map from 2009-2012. The red dots are the only locations where it was found in, well, at least 10 years ago, and we can say that at least half of the red area has already finished meanwhile. So it's a very, very rapid decline all over Europe in the last, well, 20 years, 15 years. And we're facing extinction within 10 years in the whole of Europe. We have also data from farther east, from Russia. It's also on the red list in Russia. We don't know nothing about further east, China or Kazakhstan or whatever, but it's showing a very rapid decline in its whole distribution range. I'll mainly focus on the Netherlands because our research was done in the Netherlands the last few years. And well, first of all, a map of one of the federal states in Germany. It's not the number of individuals, it's the number of populations. And we expect extinction in Germany, at least in the federal state of Hesse, within 5 years. And it was already extinct in the Netherlands in the beginning of 2000, which I will show you now. We have to realize that the decline went very, very, very rapid. We are just looking at the species, just making inventories. In the 90s, we are just had an idea about the rough distribution in the Netherlands, and within 10 years it was already gone. The blue bars are the number of square kilometers where it was found in the Netherlands in the 70s, 80s, and in the 90s you see already that half of the populations were gone. It was once widespread, but you see on the map on the right, at least for me it's on the right, the square kilometers, and the darker how the more it was found in that square kilometer. Once wide, it was, well, you could find it everywhere in the province of Limburg, but you see that the range is shrinking. You get isolated populations, then you start getting into an extinction vortex, and it's only going down, going down, and going down, and it went extinct we found the last individual in 2002 when we declared it finally extinct in the wild. Before it went extinct, finally went extinct, we trapped the last individuals from the Netherlands. It was quite shocking, but for a rodent to trap the last individuals of a rodent. Rodents are abundant. If they live there are hundreds of thousands of rodents, and we had just trapped the last 14 individuals in the Netherlands. They were really the last individuals we could find in the Netherlands. It was quite a shock to make the step and really trap these individuals. In later years, we have had some individuals from Belgium and Germany for supplementing in the breeding program for more genetic variation in our breeding program. Well, when we started, the first question was why do we want to trap our hamsters? Because at that moment there were lots of other populations elsewhere in Europe, so maybe it was easier to just go to Germany or France and trap their hamsters and bring them to the Netherlands or Hungary or whatever. But it's turned out that genetically there are very different populations in Germany. There are different recolonization lines visible in the genetics from after the last ice ages. So we have really genetically different populations in Europe. So from a point of view of conservation, it's important to conserve our local, regional, original population, which we share with Belgium and Germany. Each figure on this graph is an individual hamster and shows you in red the different populations. It's showing that Belgium, Netherlands, and North Rhine-Westphalia was the same genetically same hamster population. But you see in the upper France is different, Germany is different. So we had really a unique genetic population. Even if we look more in detail to our own population, in the middle we had sampled old museum specimens in the Museum of Natural History. We couldn't distinguish between the different populations in our region. We couldn't distinguish between Belgium or Dutch hamsters or hamsters from Germany. But nowadays, when we track the hamsters, blue were the last hamsters in the Netherlands, black were 2 German hamsters, and red is the Belgian hamster. They drifted apart. Now, at that moment when we started the conservation of the hamster in the Netherlands, we had a strong genetic differentiation between the populations. They had all drifted together to another space. Well, this is you can see here the breeding location at Gaia Zoo. Every hamster is singly housed in a large, yeah, we call it. I'm not sure about the English word for it, but never mind. Every hamster is singly housed. When we started the breeding program, we started with different breeding lines to do as much as research as possible within the breeding program. We used the breeding program to learn more about the hamsters, about the genetics, about survival, about litter sizes, etc. When we started, we had only males from the other populations. Better with Belgian population, we had only males from the Belgian population, and we crossed them with Dutch females, and we get a mixed line, the BNL line, the Belgian-Dutch line. They gave mean litter size of 4.4 with a control pure Dutch line with a mean of 5, and we had only one, the last German male from population of Noise, it's now extinct in the wild. We had the German line, and we saw an effect on litter size. There was a genetic effect on litter size, but it was just due to the good genes of the German male. There was we couldn't improve inbreeding. It was just thanks to the beneficial alleles of the German male. This was also already interesting. We also used the genetic differentiation, the unique different alleles to follow what was happening in the wild. So we used different breeding lines in different areas. So we could follow exchange between areas. We could follow how the population was doing. I will come back on it later in my presentation. But we used the genetic lines to also evaluate our success in the field, which was very important. It was possible because of the breeding line. Well, of course, a breeding program is a emergency measure. You don't want a breeding program, but if you have a breeding program, you do have to do it very, very carefully, which is done thanks to the Rotterdam Zoo and Gaia Zoo. But once there is a moment you want to release your animals again. Hamster is short living; it only lives 2 years in captivity, even shorter in the wild. So one day you have to bring them to the field. Well, you can see the field afterwards. It is alfalfa. It's a crop which is very much covered. So if you release them, they are gone. You won't find them anymore. So we needed transmitters to follow the hamsters. And well, if you use radio transmitters, you know what is happening. What will happen with your species, with your individuals which you release. It's rodents, it's a prey species, so they are eaten. And that's what we find out in the field. If you release them in the first few weeks, you will lose a quite large part of the individuals you release. But there are also individuals which survive and which reproduce and start a new population again. From all these hamsters, we have in the last few years followed more than 1,000 hamsters, captive-bred hamsters, but also wild hamsters. But we see the same pattern. We treat wild hamsters, we treat the wild offspring of the released hamsters. And what we see is that most of the hamsters are predated by all kinds of predators, which is of course quite quite logical, because it's a prey species. Its function is to be eaten. Only we want to do it. We want to have them as long as possible in the field so they can reproduce and that population can grow again. It's living in an agricultural landscape, but it's not the agriculture practice, the farming activities itself which is killing the hamsters, but it's other activities why they are created. But most of them are by foxes, mustelids, and of course birds of prey and some other unknown factors. They're sometimes eaten by eagle owls or whatever, by more rare predators. Well, this is why we kept the different breeding lines, and it's a picture with a lot of information inside. And well, when I start at the left, in blue, that's the Belgian population, and the triangle is a pure Belgian individual. The Dutch is in orange, and the Germans in green. When we started in the first year, we started a pure Dutch individuals here in orange, and we started to release individuals from the Dutch-German breeding line. They are mixed, so each point is a hamster, so they're in between, genetically in between the populations. A few years later, in 2008, we tracked again hamsters. We checked for the genetics, and we see that are mixed. So we released 2 different breeding lines in one area, but in the wild they mixed and become one mixed population. A few years later, again in 2010, we see again that there's a mixed population, genetically different. It's genetically the variation a little bit increased. So it's a stable population, and you can see that in 2010 we had no problems to find any hamsters to take DNA samples. On the bottom left, on the bottom, you see the same picture. We also released the German and the Dutch individuals, but in 2010 the population declined again. We started with a very good population in that area, the Put area, but after a few years we had a decline again, and it was almost extinct again. Unless the diversity was at a high level comparable with the breeding program. So genetic diversity was not the bottleneck within the hamster program. Well, of course, well, in the beginning we were we had the hamsters, we were looking at them and they were disappearing. But when you start looking at hamsters, you get you will see where they live, what they like. They like cover. They like a small-scaled landscape with a lot of different crops, which is also attractive for other farmland species. But after harvest, it's really at least in the Netherlands, if you then walk in the agricultural landscape, it's well, I will not say horror, but it's large. There's no cover anymore. There's no food anymore. And you can see here one lucky hamster, which has some cover from straw. But when it's gone, this hamster will be eaten in 2 or 3 days. So after harvest, it's an easy prey. It's a type of prey. It's quite heavy, a few hundred grams of meat. So for all kind of predators, it's the perfect prey. Well, we just manage our landscape in just a more or less industrial way at the moment. It's very intensively used in the Netherlands, so there's no room anymore for hamsters to live. What is happening? We looked at the population ecology of the hamster during the year. Most of the year they hibernate underground, and that's a perfect period. They have the burrow underground. They're safe. In beginning of in the spring, they start exploring for a mate, a partner, a good location to have the litter, and the reproductive period starts in May. With the first litter beginning of June, and the second litter in the end of July. But you see this lines. That is the moment when the cereal harvest starts. It's only living in cereals because it's giving food and cover. And this gray bar is the period the litter needs to get mature. But even the first litter, although it's born in June, at the moment which is quite okay, you have the time of harvest already, beginning of July, during the periods that the juveniles are becoming mature, are growing. Even the second litter, it's almost impossible at the moment. And we need at least 2 litters for a stable population. We know the survival of the hamsters. We know how much they reproduce, and we need at least 2 litters per year per female for a stable population, which is at the moment, without restrictions, impossible. So it will go extinct. You will lose at least half of your population each year. You can halve your population each year, but well, after a few years, you have no hamsters left. That was what's happening. The problem is the bottleneck is the way we manage our agriculture landscape and how we do agriculture. While we used to have horses hundreds of years ago, we now have the combines doing the same work in just a few hours. While we in the past it's weeks or even longer to harvest the cereals. Even the area of cereals has declined very much. In the Netherlands, the different cereals enormously declined in favor of sugar beets, potatoes, and maize, which is maybe good for people or for all kinds of cattle food, but for hamsters, they can't do anything in sugar beets or maize or whatever. They purely rely on cereals. So no cereals, intensification of the harvest. Well, it's almost, yeah. Well, it's showing that it's very hard for a hamster to survive in the agricultural landscape in the Netherlands at the moment. Last years we also see probably some climate effects. Very dry summers, very wet springs, and well, eroded and wet is a very nasty combination. They get ill, they get pneumonia. So it's probably not only the agricultural landscape at the moment, but also some climate change problems are also facing. The hamsters also facing some climate change. What we do at the moment, except from releasing hamsters again and again from our breeding program, thanks to the breeding program, we still have hamsters in the Netherlands. Otherwise, but we need the breeding program to have at least some hamsters in the Netherlands. The solution is trying to implement hamster-friendly management, like on the photo on the left side. We harvest the cereals, but there's still some cover left after we have harvested the fields. But it's very, very, very difficult to change the way the farmers are farming, the agricultural activity. There are so many regulations. If they have manure to put on the field, it must be done before the 1st of September. We can't pay them too much because otherwise it's not allowed by the European Union. There are so much regulations within the agriculture community because of the European agriculture policy that it is very, very, very difficult to change anything. And if we had to find something, then we could. Most of the years we had some possibilities to do it a few years, and then somebody said, "Oh, it's not possible because of that and that regulation." And it's very, very, very difficult for biologists or ecologists to find your way in the international regulations and to convince farmers to do something else. We really need a different, another common agricultural policy from the European Union. Last. Hamster is not a species which is stable on a specific field in a year. It's a species which travels through a region. So you need hamster-friendly management on a regional scale, on a landscape scale. And there are some areas where we've done it. The green areas in this area are the location where we had some hamster-friendly management. But if you know that the hamster only walks a few hundred meters as a max, you can see that even in this area, we see at least three areas. This one is, well, more or less connected, but this one more in the east, it's too far away. This is the German border, so there's no hamster-friendly management on this side of the border, but we need the Germans to cooperate. And this is on our third area, even more north. So even in our best areas, it's still not enough. There's still not enough connectivity between the fields for hamster population to survive. This was a very good year with 280 burrows. For a rodent, it's nothing. We need at least 500 individuals at the minimum for a population. So this was a very good year, but afterwards it started declining again. This graph, well, is a summary of our project. We started in 2002 with the first reintroductions. The first few years were a huge success. In 2007, over 1,000 burrows. So we thought, well, we know what to do. Then afterwards, it started. We had a crash in 2008. No voles, but a lot of predators, and they were eating all our hamsters. And now we are facing again, well, every year a further decline. Even though our hamster management is increasing, it's not enough. So where we in the beginning thought, well, we know what to do. Now we're kind of in a kind of crisis. Although we have more hamster management, we have more hectares with measures, it's still declining, and probably well, the climate is one of the factors we think that is now one of the big problems we have to face. And we really need, in the common agriculture policy, we need a fundamental change. We need more biodiversity in the common agriculture policy. So this is a lot of information in just 20 minutes, I think, 15 minutes. I'm not sure how far I am, but we've done much more, but it's more for it to read in the thesis and to ask the colleagues of Rotterdam Zoo and Gaia Zoo. Thank you very much. I'll stop sharing my screen.
Thank you very much, Maurice, both for sticking to time perfectly, but also, of course, for a very clear overview of the work that you and the colleagues at your organization, as well as the colleagues at Rotterdam Zoo and Gaia Zoo have been contributing to. I'm sure there might be some questions coming from within the audience, but we will save the questions until after all the speakers have given their presentations and collect them together. And this is also a reminder for anybody that has questions, put them in the chat box, preferably starting with a question mark so that we can easily filter them, and then we will come back to those at the end of the session. For now, thank you very much, Maurice. And we'll move to the second speaker or speakers in the session, which is going to focus on another small mammal species. The presentation will be given by Dijana Beneta, who is based at Zagreb Zoo, but many of you will also know her as the vice chair of the small mammal tag. Of course, tags have a role to play, and institutions have a role to play as it comes to our membership contributing to saving European biodiversity. And Dijana is joined by Ivan Budinski, who works for an NGO called BIOM. Dijana, the floor is yours.
Thank you. Okay, I'll share my screen. Can you see everybody?
Yes, if you can run the slideshow.
Okay, is it good?
Yeah, perfect.
Okay, thank you for inviting me. I hope you're all well and safe in this time. I want to introduce you with another small mammal. From the previous presentation, we were in the landscape, but now we'll go in the mountain. So Maurice, thank you for your nice presentation. It's really, really nice work and a lot of work. So I will contact you regarding the transmitters lately. Okay, for this presentation, I will be joined by Ivan Budinski. He is my colleague from NGO BIOM. He will be talking to you about in situ, and I will talk to you about ex situ, what we do for the Balkan snow wall. As you already know, first of all, this is not a mouse. Everybody thinks this is a mouse. This is a wall, and it's in a small mammal group. I have to tell you that small mammal tag is not that small as it seems. We almost have 1,000 members in the Facebook page, and we actually cover the species from little shrews to the giant anteaters. So you can imagine how many biodiversity we try to cover up. Regarding that, we do so many projects, my colleagues and me, regarding the financial support or knowledge support or working hours. So if you wanted to hear about conservation projects from us, please contact Tag or me or go just to the Facebook page. So back to the stone wall. As you can see, their population area is estimated at around 5,000 square meters, and it's located high in the mountain. So you can see it inhibits the area from northern Velebit. You can see the little dots where the findings are, and up to from North Velebit to the Snježnica mountain up down to the south, sorry. But as the species doesn't know the borders, they can also be found in a neighbor country like Bosnia and Herzegovina, Montenegro, Kosovo, Macedonia, and the presumption is that it's also in Albania and Greece, but we don't have any evidence about that. Just to remind you about so you can see the environment where the snow walls lives. It's really harsh environment. And it can go up to they can live up to the 2,000 meters above the sea level, and they actually inhibits the cervix and the rocks deep to the 70 meters. So it's really challenging habitat. It includes include limestone pavements on the rock surface, great karst fields, spacious karst plateaus, pits and caves, or you can name it. And it's actually a biological hotspot. So this is the Balkan snow wall. It's special because it's listed on the IUCN list as vulnerable. And in Croatian red list, it's listed as data deficient. Since it has specialized and fragmented habitats, all the population are really small and isolated. Actually, there is 3 subpopulation. Okay, regard that. Okay, actually there is 3. Sorry, sorry about that. Okay, so their population is isolated and fragmented, and they are in indirect competition with the European snow wall and indirect human action because there is development of the roads, constructions, and settlements and agricultural land on their environment. And of course, they're attacked by predators like Mustela, Vipera, or Strix. So you can all know that this little mammal is really special because it's a glacial relict and endemic species. And it's actually the only living representative of that Dinaromys genus. So we can say that we have a hidden treasure. So in 2010, the project started between Faculty of Science, NGO BIOM, and Zoological Garden of Zagreb. 9 specimens until 2012 were taken from the wild and put in the Zoological Garden of Zagreb. And in 2013, we had the first breeding success. In the second year, we already had 23 specimens. From then till now, we always have around 30 specimens, and we are very proud about that. So we put them in the small terrariums that were put 1 specimen by terrarium. What's most important to put them in the terrarium is these 3 little boxes. As you can see, they always have to have one nest box, 1 box for defecation, urination, and 1 box for bring up the food, so storage box. After that, we started to discover them, put some research. First of all, we made ectogram, of course, and then we started to explore their behavior. What we find out that voles explore the more and stay longer in the complex environment and enters the scent marked environment earlier, and female actually explore more than male. But all the behavior varies more depending on the season than the sex. After that, we took a chance and tried to find out what's vocalization between them. And what's the most important, that vocalization is highly important in this species. And they're more vocal, active during the breeding season. And actually males vocalize more than females. Yeah. Then we try to find out what they eat and how do they eat and what they do with the food. We try to bring them also the animal food, like some little insects, but they didn't try any of that. So they're exclusively herbivore. And what we find out that they eat minimal amount of food during the winter, but they prepare the food in the storage in autumn. So you have to give them more food in the autumn period. And they actually prefer dicots and leafy plant. And then we try to present them different orders. One from the conspecific, predators, and heterospecific. What we find out that male have higher latency over the predator than the female, but the females are more active in the mating season and more active than male in contact with the food. And what we find out that the specific food preference is in the mating season. So you have to think about that when they are put in the breeding positions. After that, we want to find out how their digestive tract is going on. So we find out that actually the food passes in 24 hours through the system, even though there are only 80 grams. It's really long digestive tract, but the first food starts to exit out in 4 hours. And what's important that there is no difference in digestion rate between seasons or gender. And what comes in have to go out, and we put the fecal samples in research, and we find out 20 bacterial species and 7 fungal species. What this microorganism, most of the microorganisms belongs to the soil bacteria and fungi species. And since no animal had shown any health issue regarding these microorganisms, we can conclude that they're part of the normal intestinal flora, but they can be potentially harmful if the animal is sick or immunocompromised. A logical step was to find out about their life history. And what we find out that actually they can have 5 juveniles per birth, and their gestation is more exact. So it's between 21 and the 23 days. And actually the longevity is up to 7.5 years. This picture above, it's an oldest male today. He's called Brian, and he's still living. But this season, I didn't put him in the breeding position because I think he will die. So, but I want to see how long he will live. All this research you can find out in the first doctoral thesis done by Masha Ljushtina that will be on available soon enough. So what we know about them, we know that they are active through the entire year, but most often at dusk and at night. They're very agile and fast. they're actually jumping. Their jump is more like a combination between a squirrel and a goat. So you can imagine, but it's a wall. And they have glands and mark the territories by the secretion. And as I said, vocalization is a really important part of the behavior. So they're strictly herbivore. You have to put them in various types of plants and grass, and browse is most important because of their teeth. And they store food in their nest or hideouts, and they have seasonal diet change. Their average number of juveniles are 2 to 3, and they have actually up to 2 liters a year. They reproduce through the entire year, but we put them in the breeding position in March and in October. And they actually reach the sexual maturity during their 2nd year of life. That's really long enough, because in the nature they live about 2 or 3 years. Females carry the pups around 30 days, and they actually build nests under the rocks on limestone and small caves. What's important that in Dinaromys, love hurts, because first contact is always aggressive and always is female that is indicate this position. So she will go to the male territory and attack male. But at the end, they can, love is blossoming. And you can see the progression. This is actually a pregnant female that gave birth that night. And you can see development of the juvenile from the day 1, day 7, day 21, and day 28, when they start to exit the nest and bring up the food in the storage. So with all that knowledge, last year we put a new enclosure for the visitors with all the elements that they need, and that we have information from the wild, and it has a great educational value. For the future research, this is the list. For the baseline, we will try to find out the baseline hormones in the males and females. We already have the samples, and ongoing is skull morphology, histology, and digestive system, digestion rate, and you can see what we plan to do with them. And now I will give a floor to Ivan, who will talk about in situ. Is it okay?
Yeah, okay. I'm here. Now I will share the same presentation from my.
Ja, wegen dem Termin haben Sie mir eigentlich gesagt, dass jetzt da alles in Ordnung ist. Das heißt,
Can somebody please mute for the room?
Vielleicht, wenn du irgendwann mal sowas machen darfst.
Okay, can you see your presentation?
Yeah.
Okay,
Please go ahead.
I'm here to represent some in situ research for Balkan snow vole, because the species was previously known from only a handful of sites and very small observations, and it was trapped only occasionally while other small rodents were researched. This is another picture of a typical habitat. But
Ivan,
if you look,
Ivan, sorry to interrupt you briefly, but is it possible to put on the slideshow so that we have a slightly better view of your presentation?
okay, just a second. Yeah. Yeah, this works.
Perfect, thank you.
Okay, so if you look at the habitat, this would be a typical habitat, a mixture of rocky limestone outcrops, some forests, so shrublands, and some rocky grasslands. But only in this rocky outcrop in the left side is something that the species uses as a habitat. So all surrounding habitat types are not really used, and it's almost never registered there. So that's one of the reasons why the wild population is fragmented, because they are really using some type of habitat that's fragmented. This is another picture from Montenegro, from Durmitor Mountain, where there is a large, vast areas covered with karstic, with limestone grasslands, dry grasslands, but only this in the middle, look like a small scree, like a pile of rocks, is something that this species uses. And also in understanding the species we were also helped by research in captivity, because the species really avoids to stand on any soft surface. So it will keep its feet on rocks. As long as there is some dry grass or leaf litter, the species just avoids to step on it. We consider it as an anti-predatory behavior, because as long as it moves on rocks, it's completely silent. When it starts moving on some leaf litter or something like that, it becomes, you can hear its movement, especially for predators. But that was very nice connection about ex situ and in situ research. So we also knew that the species is very probably trap shy, because when we were trying to trap first specimens, especially those for zoo, for ex situ research, we were aware that even in a site where we can observe some signs of the species presence, we couldn't manage to trap it, even in a very small area with a lot of traps. And in some cases, the first animals were caught like after 3 or 4 days after trapping in the same micro location. What was again shown in captivity, the species is also very shy, new object shy. So understanding when you put a trap in its habitat, usually like for Apodemus mice, which are very common in Europe, they would approach a trap very fast because they're inquisitive. They will try to investigate what is that. But for Balkan snow vole, that species will not approach very often the trap for first 2-3 days. And because of the standardized trap-based small mammal research is usually fixed on 48 hours on a location that's very often just too short for a trap-shy species to enter a trap. So we probably have lost a lot of distribution data just because of that 48 hours that is usual methodology for keeping traps on each location. So we decided, because in the same time we have also started a lot of other mammal research using camera traps, photo traps. So try to use them in those crevices with some perception where the species should be present, and the results were very good, because we managed to observe or to record the animal even in sites where it was reported that it was extinct, or in sites where we couldn't trap it using camera trap, just using normal traps, cage traps. So the camera traps really worked great. But the issue is that camera trap is usually used for larger animals, and is used in a way that's mostly fixed on trees. So we have to invent. We have to start doing it inside the crevices, something like very, very small caves, large enough for camera to be put in, and because the animal is using the Balkan snow vole is using such in crevices, and it worked really good. Of course, for a specialized small mammal research like this, you need a mammal that you can easily recognize on a camera trap. So, but Balkan snow vole is mostly very recognizable. Only in some cases, if the photo quality is low, or the animal is in far distance from a camera trap, it can be confused with common snow vole. But in most of the cases, it's clear that this species is present. But we also did observe some behavior that's also interesting. This is one footage of how to search for those crevices and how we are working that to putting traps inside. And this is also one slide of me trying to fix a camera trap inside some of those crevices. The good thing about,
I'm really sorry to have to do this, but
I'm almost finished.
Yeah, 3 more minutes so that we make sure we have time for the other 2 talks as well. Thank you.
Okay, thank you. This is another. So you see on the left photo, you see how the camera is fixed for a rock to take photo of inside the crevice. And usually this is the type of footage that we get from the wild. So somehow we usually call it like a small cave, because for an animal like a Balkan snow, it's a small cave, and they tend to spend a lot of time inside, although it is much larger than them, so they are not squeezed inside, but they prefer, they feel very safe inside those caves, so they are very often cleaning, just resting, looking around, or in some cases jumping through. We also do, according to their behavior, trying to decide, is it the permanent territory? So it would be like bringing some food for cache or breeding nesting material, or if it is some dispersal area, so like the area that the species is present only in when some floaters are foraging and looking for a new territory. And of course, during the research, we've developed monitoring method for, the species is Natura 2000 species, and it was included in the list by Croatia, EU list by Croatia, but no one really knew how to monitor it before, because it's so much trap shy, and its observations are very scarce. We also did some promotions, and that's cooperation with Zoo in Zagreb is also part of promotion. And that's it. Hope I didn't, I followed the schedule and minutes allowed to me. This is not in photo from the WAD, this is photo from the Zagreb Zoo.
Thank you very much, Ivan, and also Dijana, for your wonderful presentations. And I could easily sit and watch you talk about the species for another half an hour, but I need to be a bit strict with time, because we only have 10 minutes of flexibility, because there's another session right after this one on this Zoo stream. So thank you very much. Again, if there's any questions, I see some in the chat already for Ivan or Dijana. Let them know, and maybe you could already start answering some of these in the chat, because I fear that we might not have more than 5 minutes of questions at the end of the session. So that might help the participants that are asking questions get an answer. Thank you very much. And then the next speaker, looking at an institutional EAZA member angle and what you can do as a European wildlife park or an EAZA member with a European collection, is Anthony Kohler from Grotte de Han on Lesse. Anthony, it's up to you. Go ahead.
Hi, everybody. I will share my presentation. Oops. Okay, it's okay. Everybody see it?
Yes.
Okay. The idea is to have a look of how zoo can contribute for conservation. We can see on these two posters we can do a lot. And yeah, when we see this poster, it's just the visible side of the iceberg, because yeah, it's only 100 members who or give the information of what they do for the conservation. So it's very important for everybody to put the information in the conservation database to have an idea of what we are doing really on the field. What strikes me when I see this poster, it's only 14% of our global effort for conservation is about our European territory. Yeah, it seems to be logical, because during long time the conservation in Europe was not seems to be a very suitable place to make conservation, because of our way to make agriculture, forestry, or because of the urbanization. But we are living a very nice moment with the idea of rewilding Europe, and we try to restore some natural missing process, recreating richer ecosystem based on the reintroduction of large animals to conserve not only the species, but also to shape a new environment with free evolution. And if we do that, we can lead to come back the lost natural process. And what is very interesting, it's only one single species could modify very the landscape. And when we think about it, we all think about the bison, because the bison is the emblematic species of the rewilding movement. Nowadays, we talk about 5,000 European bison in the wild in Europe. But we have a lot of small population with not a very long-term viability. We need more bigger viable population, and for that we don't have finished the work to reintroduce this animal in the wild. And we don't have to forget bison as so rare as the black rhino in Africa. And we talk about black rhino, but it's the same with this bison. The project about the bison have two objectives. One is, of course, the protection of the species, and the second one is to restore the ecosystem dynamics through the bison function. And I want to present you one project. I have been very lucky to go in Romania in 2015 to release the bison and to follow what happens there. This is during the rewilding bison action plan. They plan to reintroduce bison in the Carpathicus mountain. In the south of Romania, we have the mountain of Tacu. This is a project with WWF Romania on rewilding Europe. And what is very interesting with this project, it was a request for the municipality and the resident. And when I go there, I was very impressed by the support by the population. You know, you are driving with the trucks, with the animal, and all the people are outside in the street to welcome the animal. And we have a meeting time with all the population for the release of the bison, but the road was not very nice to go there. So we are in late, 4 hours later to go there, but the children, the population stays there to welcome the animal. And it was a very important moment for this population to say hi, to come back to this animal. And we know now we can't save a species without including local population. And when this is an ask of the population, yeah, the success is guaranteed for that. Of course, most of the people were there to see the release of the bison, and it was a very great moment also for the media. We have a very strong enthusiasm about this project, of course. And it was all over Europe. We talk about the comeback of the bison. And after that, we can talk about a very success story. There is 2 live projects about this particular area. 64 bison have been translocated in this project, and since 2014 it's more than 88 animals. We have also a stable population now in this area because we have natality and mortality equally around 20% of it. The animal use a landscape more than 94,000 hectares. And we have a very nice record with the highest point we record the bison in Europe. But what next with this project? It's the project want to continue to support the local community. It's a key part of this project, developing the infrastructure to boost the ecotourism in this area. They start to develop this ecotourism to see the bison, to sleep in the area. It's like this. There is more financial potential in this area for the local community. It's also generating opportunities for sustainable business and creating jobs, encouraging more young local to remain in the area. We also see this kind of result with a lot of ecotourism about wildlife all over Europe, even with brown bear, for example, in Slovenia. It's help young local people to stay in the area. And of course, it's not just one-shot initiative. It's a long-term, spanning over 25 years to continue to expand the future population. And in Romania, they can have the more viable population of bison all over Europe, because we have a very nice connected area all over the Carpaths. Like this, they can have a very good improve about the bison population all over Europe. But bison, it's very nice species, and many people want to collaborate, to be involved in the bison conservation. But we can also help some more smaller species, as we can see with the hamster, with the vole, but also with some reptiles like the smooth snake, Coronella austriaca. In Belgium, we have the more also the biggest population all over Europe, with the more concentrated population, and the population was growing, so everything was good. But we have a work about the rail for the train on the largest population, around 300 individuals. And of course, they have to destroy all the habitat. So we have to think about a solution to reduce the impact of these works. So the idea was to create a center during the works to collect the animal the most as we can, put them in this center, and after to release them in the wild if it's possible. So in Sur-Lesse, we create financing and take care of the center. And we take in charge the animal before they are returned in the wild. So it was a very big challenge, because how to buy an enclosure for the smooth snake, it's very difficult, because we have not so many experiences with these species. We know they can easily escape. They need to hibernate in a naturally way. So we have to create some specification with some expert to see how we can work with them. We need 10 enclosure of 25 square meters for a capacity of 100, 150 animals, because we know we can't save all the population. It's too late to have the time to save all the population, because in idealistic way, we need some 3-4 years to do it, but in the realistic way, we have less of 2 years to do it, so it's impossible. We have to think how we can manage the population in each enclosure, so it's around 10 or 15 animals, and more females than male if it's possible, to reduce the competition for the male during the mating period. Since the start of the project in 2017, we saved 181 snakes from 3 populations in Wallonia, because they will continue to work on all the rail train to improve their infrastructure. We have to manage how we can save most as we can animal. So for the design of this enclosure, what is very difficult, it was to not let them go outside. So we use simply a gutter like this. They can't climb or escape. We need a grassy area for them to hunt, and we learn very quickly if we let the grass grow too high, the snake can just climb on the grass to try to escape. So we put some rodents in the enclosure to get the grass on like this. With guinea pig, it was more easier to manage it. We need also stony area for the shelter, for the hibernation of the animal, and we create this thinking just, okay, we put the animal inside, we see what happened, and after we catch them in 5 years to release them in the wild. Yes, but in case we want to see how they adapt in this enclosure, so we have to use some plate to let them go outside to just to take their temperature. And we make some capture markage. We capture with the animal because they have a particular task behind the head. So yeah, it was more work that we expect at the beginning. We also have one big challenge is to pass this animal with they are eating in the wild some slow worm or lizard or something like that. But here we have to put food with some baby rats or baby mouse on. Yeah, we are very lucky because it works very well with them. The other challenge was about the birth management, because with this kind of enclosure, it's quite impossible to find the young in time to release them before their 10 days, because we want to release them as soon as we can, to let them the time to explore their new environment before the winter. So we catch all the female in mid of July to put them in little box. After we wait for the giving birth. When they give birth, of course, we separate the young with the female. Just after the birth, we of course record everything we have to record. And the mother is placed in the outdoor enclosure after a few days after the giving birth, just we have the time to have a health checkup, because yeah, it could be a tricky situation after the giving birth for her. And after, what was not planned at first, it's to reintroduce the young, because we do not expect so many young. So it was a very surprising situation. So we have to find in emergency 3 sites of reintroduction. We use 3 since 2016. One was, they are all in the favorable site located in the range of the species, with thermophilic environments, with slow worm or Lacertidae, on many shelters for them. And of course, there is no small snake in this area, just to create a new population. The first site, it's created with 20 hibernaculum on the more than 30 scree beds designed for reptile over several kilometers. And we release in this site 129 young. We have the obligation to do it because it was there is always a population in this strange area. It's a big, it's just between a trail lane and the auto road. But yes, since long time there is a population there. So yeah, we have to reintroduce animal here. We have also a carrier in activity with a very large natural area, lot of prey, no smooth snake. We released in 2017 162 animal, only 61 animal in 2018 because we have less births this year. In 2019, 254. On another site with all carrier with it's now a natural reserve. There is a lot of prey, no smooth snake also. We release this year 135 animal. About the smooth snake in Belgium, we partially saved the exceptional smooth snake population. We are quite happy with the breeding operation. It's yeah, a success. We find new information about the species, and we because in the wild many female give birth as most as they can young, and they are not really able to give birth the next year. Here they can give birth each year, less young, but each year they can breed. So it's not the same strategy for her. We collect, of course, new data about the reproduction of the species, precocity record for the birth, record average for the litter size. The record is 16 young for one female. But the compensatory sites are too few and settled too late. There is not enough time for the colonization by prey. So here we do a lot of effort for this species, but it's quite difficult to have the compensatory sites ready in time. About the reintroduction, we need to follow it more deeply in the future, because during the first year of the species, it's quite difficult to contact them in the wild. So we choose to wait during 3 years and after to have a monitoring more deep to see what happened really. So I hope next time I can give you some good news about it. Another species.
Anthony, you have 2 minutes left.
Yeah.
Again, very sorry to have to do that.
No, it's okay. It's okay. I will do it very quickly. It's the same for yellow-bellied toad. The situation in Belgium is a declining population, critically endangered. The idea is to make restoration of his habitat in the wild, and we're breeding in ex situ project. So we have a breeding center with 10 adults. We manage them in terrestrial terrarium, easily feeding for the crickets, hibernation in around 4 degrees in a fridge. And for the breeding, we have a specific water terrarium to stimulate reproductive behavior. Like this, the female don't really have the choice, but they can escape if she wants. We collect the eggs, isolate for the eclosion, and we isolate the tadpole in little box by 100 animal. 3 breeding sessions for this year. And after we have two ways to breed them and to manage the young. Indoor with 25 animals, no more because of chemical competition in the water. You know, they secrete growth inhibiting hormone. We can boost them in 4 weeks, but there is more mortality. With the outdoor, we can keep them by 100. In 6-8 weeks, they can be ready for release. Few mortality. We release them in active carrier. No species was not present. We make a lot of arrangement for them, and we release 1,088 tadpole this year. We have to optimize the production of egg for the beginning and the end of the breeding session. And we have to wait for the result of the reintroduction. But with this reintroduction, we create the second most important population in Wallonia. Just to finish, there is real need about conservation in Europe, a lot of opportunity to help, not necessarily a big and expensive project. Many species are endangered in Europe, European mink, wild forest reindeer, bird vulture. There is more than 400 species critically endangered on the red list. So yeah, help us to make Europe a wilder place. Thank you.
Thank you very much, Anthony, for that overview and these 3 species examples. And I know these are just 3 examples of many more that you're doing. So that's really excellent. And I see that Dijana and Ivan are doing a great job at answering the questions that are asked in the chat. So, there are a few for you as well, Anthony, so maybe you can do the same. And Maurice, there's also several for you. If you can scroll back maybe to a bit earlier in the chat, and you can maybe start answering some of the questions on the common hamster. That might help us in terms of time. And maybe to give you some pointers, Maurice, most of the questions related to the rapid decline or the fast growth and then the rapid decline, and questions as to whether they are linked to predator populations, whether chemicals have a role to play, and what this means for the ongoing continuity of the reintroduction program. So if you don't mind, Maurice, to also feedback a little bit on those in the chat, then we might just make it in time. Without further ado, I'm going to introduce the last speaker, Elmar Fienieg, my colleague from the EAZA office, who will talk about a desk study work that we do looking at ex situ conservation in light of the EU legal framework for conservation. Elmar, over to you.
And I found the unmute button.
Very good.
Yeah, that's always handy. So it's good that everybody has already explained a lot about what ex situ conservation is, and so I can skip that definition, and we can gain some time. So you've had a lot of talks already about European ex situ conservation, and you may have started wondering what is actually happening in Europe, and how many of these actions are happening there. And this is exactly what Danny, Clara, and I were also thinking. And then the Life Fund provided us with the money to actually look into this and investigate this. So that's what we did. And those results I can share with you today. If my slide would, yeah. All right, so for a bit more background, so the European Union just launched its 2030 biodiversity strategy, and in it, it says that it wants the European biodiversity on the road to recovery in the next decade. And for that, it might need EAZA. So that's quite convenient, because EAZA also would like to contribute to the conservation of European species, because it fits in our mission, progressive zoos and aquariums, saving species with you. Now, if we look at Europe, then we have a pretty good idea of what species we are talking about. Those are the ones that are listed on the EU Habitats Directive, Appendix 4, and the EU Birds Directive, Appendix 1. So those list the species of the highest conservation concern of the European Union. It's about 575 species. But then if we zoom into the with you part, we don't have a really good picture. So as you've seen, many EAZA members are involved in ex situ conservation projects, and of those we are kind of aware. However, beyond the EAZA, we did not really have a good overview of what was happening. So that led us to what was practically our question: who's working on what ex situ conservation actions and for what reason? Now, while we were investigating this anyway, we immediately wanted to see if these ex situ conservation actions were also following a one plan approach in their planning part, and if they were following the ex situ conservation guidelines. So we wanted to know if they were an integrated part of a broader conservation strategy, and were they engaging with all relevant stakeholders? Were all roles and aspects sufficiently considered? Now, if you want to answer these questions definitely for each project, it would require a lot of interviewing and a lot of time, which we did not really have. So instead, we chose to look online to see what information we could find on ex situ conservation projects, and could we learn something about these projects in line with these questions. So that brings us to the methods for data collection. So what we did was practically look online for each of the 575 species if we could find any ex situ conservation actions that happened in the last 10 years. And if we found them, could we link them to a broader conservation strategy? So especially Clara here did a huge amount of work, and went a little bit already in a lockdown with staying months on the internet, searching in every little piece of the internet, every database that was out there, or everything Google had to offer. And eventually that allowed us to end up with a nice big spreadsheet, which provided us a bit of an overview of what was going on. So what could we learn from this overview? Well, we found 287 unique projects, and those were involved in 473 ex situ conservation actions. So because some projects were working on multiple species, we have more actions than projects. This covered about a third of the investigated species, so almost 200 species, for which we had actions. And in reality, this proportion might actually be still higher, because even though we really did our best to find everything online, there must be things that were still hidden behind language barriers, or if there is no information on the internet, we could also not find it, of course. So this currently includes rehabilitation actions, which generally are quite small scale. So we thought it was also only fair to show this number without rehabilitation actions included. And if you look at it then we get about 22% of the species for which we have actions. If we then look at the different species groups for which these actions we're aiming at, we see that in absolute numbers, we have a huge number of actions for mammals, birds, and amphibians. And relative to the number of species that we actually investigated, we actually found a lot of actions for reptiles and fish. Now, for terrestrial invertebrates, there are a large number of species that we investigated, over 100, but we actually found relatively few actions. So what were these actions trying to achieve? What was the purpose? What was their role? Before we dive further into that, it's good to remember that an ex situ conservation action can contribute to conservation directly or indirectly in a variety of ways. So this is why I have this nice colorful slide here to show you the diversity. Reality was a bit less diverse. Most seem to function as a source to establish new populations or demographically boost existing ones. And you see a bit of rehabilitation here as well quite a large green surface, but that involves indeed a lot of actions, but only 13 projects that worked on a very large number of species. So when we look at it proportionally, it would be only 5% of the actions. Now, there's a lot we can learn from this, and we do not have time to discuss that. But one thing that we thought was particularly interesting was the low number of projects that were aiming to increase genetic diversity of the wild population, so a genetic rescue. So we know that there's actually, well, there have been several scholars which have argued that there's quite a high need for genetic rescue. So then the proportion of projects seems quite low. So who was leading on these projects? Were they just a few organizations that were tightly linked together? Well, not really. We actually found 151 unique leading organizations. And you see about, if every circle here, every blue circle represents one organization, then it should be about 3 times the number of circles shown here. And these were a lot of regional governments, but also national governments, zoos and aquariums, of which most of them EAZA members, NGOs and universities. Now, every project also had an average of 1 to 2 formal partners, which involved an additional 300 unique partners. So in total, we ended up with 450 organizations that were involved in these projects. And some of them were linked together, but many of them were not. And in the most extreme case, we found, for instance, 20 projects that were working on the same species in different areas, but none of them listed any of the others as a formal partner on the website. So we have some cooperation, but in general, we have a very diverse community working on European ex situ conservation, but also seemingly a fragmented community. So were these actions a broader part of a larger conservation strategy for the species? So did they fit nicely in one plan and fitted nicely with the other actions that were ongoing? Or were they individually planned and without really knowing if that actually fitted well with any of the other conservation activities that were ongoing? Well, what we found was for about 29% of the species that had ex situ conservation actions, we could link them in some way to a higher level plan or at least a higher level authority. I must add here that this was quite our most optimistic interpretation of the available information. Probably a more realistic answer to this question is we do not really know what the ex situ conservation needs are because there are too few species conservation action plans published or at least accessible online. And the plans that we did have were often lacking sufficient detail to determine ex situ conservation needs. So what do these results tell us about the road forward to recovery by 2030? Well, it seems that there are a few cracks in the road. And with few higher level plans, it's quite unclear if ex situ conservation is needed and why. And that makes it quite difficult for EAZA and many other organizations to plan their contribution to European conservation. also in this diverse community, but quite fragmented community, we cannot really talk about an established network, it seems. And without a network, there are relatively few opportunities for collaboration and knowledge sharing. Now, knowledge sharing actually seems quite important because, as we know, ex situ conservation is complex and involves a lot of aspects, and no one is an expert in everything. So there might be some missing knowledge in some of these projects. So how can we improve this? How can we get more European-wide species conservation action plans, especially including clear ex situ recommendations that are specific and consider all possible roles? Well, if we look at the plans that have been published so far, they generally involved support from the European Commission and from the IUCN Conservation Planning Specialist Group, and from BirdLife. So if that was a successful combination for those plans, it might be for the future ones as well. So it seems to make sense to build on forward onto that relationship, and for these parties to talk together and see how they can take action and find a way to ramp up development. Now, whenever this would involve an ex situ component, it may also make sense to include EAZA in those conversations. For instance, to give input, but also to see how this may fit into our regional collection planning process, which is now a lot more flexible with our new population management structure. And how can we improve the network that is out there? Well, we have these hundreds of organizations that are working in tens of countries in Europe. When you listen to that, it actually sounds quite familiar. Actually, EAZA is already such a network of hundreds of organizations working in tens of European countries and others. So there seems to be a bit of an opportunity there. EAZA is already well organized and has a huge amount of expertise and knowledge. So right now, there are people working in our own backyard that do not have access to that knowledge. So maybe there's some room for improvement. I do have to add there that around 90 of these projects already have EAZA member involvement. So in a sense, they are already part of that network, at least in some way. But for many of the others, that is not the case. So yeah, how do we move forward with this is something that we have to figure out, and probably does not take a one size fits all approach, but more a tailor-made approach for many different types of projects. So what I would like you to do is to think if you in your position in EAZA or in your organization can in any way improve the situation with collaboration with any of these projects, and share them with others, or for instance with me. This is why you have my email address. So that was it. We hope to publish the results of this study later this year, and for now I'm happy to take any questions if we still have a bit of time left. Thank you everyone for listening.
Thank you very much, Elmar. There are indeed a couple questions to you in the chat, so if you can look those up and maybe answer them there. That allows me to ask one question, and then after that I'll wrap it up, and I'll ask the question to Maurice. Maurice, you talked about the complexity of the rules at the EU level, at the national level. And that makes the work you do for hamsters, but it's probably not unique to hamsters in the Netherlands or even beyond the Netherlands, complex. Do you have any thoughts or suggestions of how that could change, or what would be a single simple thing that might make that a bit more efficient for the work you do?
Well, we have, of course, within the Common Agricultural Policy, the different pilots, the pilot 1 and the pilot 2 and the pilot 3, etc. And there is also the the greening measures for farmers. They have to implement some greening measures. It's totally focused on a healthy environment, but it's not focused on biodiversity. If you can at first change the greening measures into more biodiversity friendly measures, that would already be a step forward. Of course, we need more, but that's already a first step. There's now some greening measures are meant to support the environment and nature, are in practice the opposite. We had some really nice hamster fields which were used for greening measures. Well, all hamsters were gone in just 1 year. So let's start with the greening measures to make them really biodiversity friendly.
Right. Yeah. So put biodiversity at the heart and at the core of the decision making is I guess what the essence is of that, and I would agree.
Yeah, and they have really no idea in the European Union or at our own ministry. It's just there are walls between the biodiversity management, the biodiversity sector, and the agriculture people. They don't talk with each other. They have no idea. We should knock on the door and get them out and show them what the effect is of their plans.
Yeah, no, that's very clear. Thanks so much, Maurice. And we have 4 minutes before we kicked out of the stream, so I will quickly wrap up the session by thanking all the speakers for the hard work and the enthusiasm as clearly shown in the work you do. I think from across your 4 presentations, we are showing a quite nice diverse range of activities in different sectors on different species across the European Union. I really picked up that husbandry expertise is something that is key to the work that we're doing, and that's where our community has a lot of success. We can definitely also be a partner, a contributing partner to initiatives that are led, for example, in the case of the hamster by universities and other organizations. And we definitely have a strong network across Europe as an association where there is scope for more organization of executive conservation work, as Elmar has showed, and we have the ambition to do so. So with that, thanks again to the speakers, thanks again to the participants for being active and asking all your questions, and I look forward to see you in the next sessions. Thank you so much.